So I'm here to talk to you about genome editing, how, what, and why. And I feel really passionately about this area, not just because of its applications in medicine or in genetic understanding, but because of its applications among every single field we know. So I would love to tell you a little bit about how it works and where we might see its application in future. So my disclosure, I am CEO and founder of Fertility Health, but there is no conflict of interest there with this. So CRISPR genome editing has been referred to as genetic surgery, molecular scissors, a find and replace tool. These terms that we use to explain genome editing go a long way to describe its applications. Talking about it as genetic surgery is a really great way of understanding how it could be used essentially as a very fine tool. When you look at some of the current tools that we use, actually surgery probably doesn't go a long way in saying how accurate it could be on a molecular level. So maybe using the word molecular scissors or a find and replace tool are a little bit more appropriate, but you will see these expressions used interchangeably in the mainstream media. So since its initial kind of seminal paper in 2013, this has dominated the headlines for not just Nature and Science, but also global headlines in terms of the mainstream media. And it's no real surprise. It has really revolutionized many of our ways that we can adapt, many of the ways that we can understand and appreciate biology. So you can see really quickly from this diagram here, or from this graph, that since the seminal paper in 2013, there has been an explosion. This actually doesn't show the full appreciation. There are now over 20 million search hits for the term CRISPR when you Google CRISPR. So what I'm going to try and explain to you tonight is essentially a snapshot of the technology where it is now, but it is really like trying to explain trying to take a picture of somebody who is running. You get an overall impression that what is happening and you know they're going fast, but truthfully, this technology is moving so quickly that even by the month, some of the techniques are becoming outdated. So I really think that's important to remember. Even in the 1 year following the seminal paper by Emmanuel Charpentier and Jennifer Doudna, where they applied CRISPR to genome engineering, it has been used in almost every other model organism there is. And I always think that this is a real testament to a technology when it is easily replicated and reproduced, and results are successful in the hands of any scientist. And this is certainly the case with CRISPR. But what is CRISPR? People often think that this is a new, strange, or foreign phenomenon, and it's a new technology. And yes, it is a technology, but really what this technology is harnessing a very simple, very old technique, which is a bacterial immune system. So CRISPR itself is not a new thing. CRISPR is an ancient immune system used by bacteria in order to fight off invading viruses or other bacteria. So it's actually quite nice to look at the process here. We often remove ourselves of the physical entity of what is happening, but this picture shows a phage landing on the surface of a bacterium. And as you can see from this phage, it's actually physically inserting its DNA into the bacterium. You can see it down here. And this is an electron microscope photo of this happening. And what's wonderful about this, as you can see, the phage inserts its DNA and then just flies off. What's wonderful about this is the fact that when you understand that actually a virus, once it inserts its DNA into the bacterium actually has an immune system that fights this off. And that immune system is an adaptive immune system called CRISPR. So it would be remiss of me not to explain what the acronym is. It stands for Clustered Regularly Interspaced Short Palindromic Repeats. And essentially what CRISPR refers to is the genetic sequence in this bacterial genome. So this can be very easily broken down and be easily understood if you actually just say the words a little bit more slowly. These regularly interspaced short palindromic repeats were found clustered throughout the bacterial genomes. And they were very elusive in that scientists didn't understand what these sequences were and what was the interspaced sequences in between. what we've uncovered is that these interspace sequences actually served as a genetic historical timeline of invading bacterial genomes that had been inserted into a bacteria's genome. In other words, it served as a historical timeline for every time a bacteria had had an infection from a virus, fought that infection, and won by inserting that piece of foreign DNA, thereby rendering itself immune to any further infections of a bacteria of a similar genetic sequence. So when we talk about CRISPR, we always say CRISPR-Cas, and really the Cas stands for the endonuclease, or what I was saying earlier, the molecular scissors. It's essentially just an enzyme that will cut the DNA. Now, typically, we always refer to CRISPR-Cas9. I always think Cas9 is like the playgirl of the CRISPR world. It was the first endonuclease that was used. She was quite promiscuous, was known for cutting all sorts of places in the genome. But actually, since the discovery of Cas9, hundreds of other bacteria have been sequenced in order to find more accurate, more precise, and just other alternatives for Cas enzymes or endonucleases. And actually where we're going with this is not just using a standard endonuclease, we're using synthetically altered enzymes that allow us to have a greater packing capacity size-wise. Sometimes Cas9 was too big to get into the cells. So we're really evolving in both our tools, but equally the endonuclease that does the cutting. But it's very important to think of these two components because both of them are very useful in genome editing and beyond. So just to show you a quick example here, we can see the phage landing on the surface of the bacterium. And as it does, as I mentioned, it inserts its DNA into the bacterium. When the bacterium then detects the presence of the virus DNA, it produces two types of short RNA, one which contains the sequence that matches that of the invading virus. And these two RNAs form a complex for the protein Cas9. As you can see, this big rock-like sequence in the background. Cas9 is the endonuclease, which is a type of enzyme that can cut DNA. When the matching sequence, known as the guide RNA, finds its target within the viral genome, the Cas9 enzyme cuts the DNA, disabling the virus. Once inside the nucleus, the resulting complex will then lock onto a short sequence known as the PAM. And this Cas will unzip the DNA and match it to its target RNA. So this process here is really, really adaptable to where we can actually not just use this find and replace with these guide RNAs, but also the cutting. As I mentioned, there's two separate processes here that we need to think of. But importantly, as soon as you break DNA, DNA wants to repair. So you'll all be quite familiar with the two types of DNA repair. One is non-homologous end joining, which is very quickly and often referred to as the error-prone method of repair. I think it's probably not as error-prone as we give it credit for. Our cells are constantly repairing, and if it was that error-prone, we'd all be a bit more error-prone. Nonetheless, it is error-prone in the sense that these ends just quickly want to join together, and sometimes you will lose base pairs here and there. Now, for the purpose of an experiment that just wants to knock out a gene, you don't really mind whether or not a few bases are missing. However, if in DNA you were missing a gene, it would be a favorable experiment to do homology-directed repair. So the other type of repair that DNA undergoes is homology-directed. In other words, and this usually happens when cells are dividing, so there's plenty of DNA transcript around for it to enable a correction of that break. So it actually really matters to your editing outcomes what kind of repair you are looking for and what kind of edit you are trying to make. But the idea of doing genome editing with engineered nucleases is not a new one. In fact, there have been many predecessors to this technology that have adapted and tried to use forms of molecular scissors and programmed nucleases, such as engineered meganucleases, zinc finger nucleases, which you'll quite commonly hear about, and transcription activator like effector nucleases. And while these are successful to an extent, they are clunky, and I would essentially call the editing outcomes comparable to what you would achieve on a typewriter. Yes, you can rewrite code, but if you make a mistake, it's very expensive and clunky to refix. And they have been used in the clinic, and we're seeing more and more cases where companies are releasing their phase 1-2 clinical trials using these former methods of genome editing. And the reason is because they've gone through the phase 1-2 clinical trials. There is not one of those companies that is not adopting CRISPR as a replacement for their forthcoming technologies. Here is an example of where genome editing was used in a young girl called Layla, and she had a type of blood cancer whereby she had no white blood cells left, and she had undergone every treatment there was. She was given an experimental license to do genome editing on her, and 5 years later, she is now happy and healthy. So there have been incidences where people have actually used an emergency experimental license on humans for somatic cell therapy, or they have opted for them, such as the man on the right with Hunter syndrome. So a quick run through of how somatic cell therapy works is to treat disease in their somatic cells, so the cells that live and die with them. How this broadly would work is that you would take the cells of the actual sick individual themselves, you would edit them to correct the mutation that they have, and then you would grow these cells up and retransfuse them back into the patient themselves, thereby foregoing any need for immunosuppressants. You reduce the risks of graft versus host disease. You're using the patient's own cells to cure their own condition. Obviously, this is much more applicable to some types of disorders and diseases, especially blood-based disorders and cancers. But preclinical studies in non-human primates have shown a single dose effective use of base editing, which actually showed that they were able to permanently reduce LDL cholesterol in patients who suffer from familial hypercholesterolemia. So I really think this is going to make massive advances in the clinic, whereby you can actually do a single dose and it's sufficient enough. And I think threshold is a really important thing to consider. The sufficient enough editing change to enable a recovery in a given patient. Equally, there have been some mouse animal studies that have shown potential for fetal gene therapy, doing in utero gene editing to correct disorders that have been detected in utero, such as spinal muscular atrophy here. But what if you are to try and prevent genetic disease? Among the IVF industry, I think it's quite a familiar idea that you would prevent an aneuploidy, but when you come to prevent genetic disease, it's quite a familiar technology to use pre-implantation genetic diagnosis. However, applying genome editing to prevent genetic disease invokes all sorts of negative connotations. So first of all, before we talk about the application of this technology in human reproduction, I think it's perfectly placed to just take a little bit of a time to look at the current treatments that we use for treatment of genetic diseases, such as pre-implantation tissue typing, pre-implantation genetic screening, diagnosis or PGT-A and PGT-M, mitochondrial replacement therapy, which has been granted a license to be used going forward, or else you have the option of a use of a donor. And actually, almost every one of these, barring the donor, could be replaced through successful genome editing technologies. But we'll discuss the logistics of that. But I think when we look at the image underneath, or more clearly here, if we were to integrate genome editing into assisted reproduction, very often people assume that this is an entirely different field, or that it's a gross aberration of the current practice. And in fact, there's very little difference to what we're currently already doing when we manipulate an embryo by injecting a sperm into it. Possibly that is the exact same time that we would inject the nuclease in order to create the edit. At the exact same thing, we would wait for the embryo to grow. The cells would be tested to ensure that they then did not carry the mutation that you were trying to correct, and then they would either be vitrified or transferred for pregnancy. So when we usually talk about any of this, unfortunately, the dominating headline is always about designer babies. And this is a very unfortunate thing, because it really does take from the unmet need potentially in given cycles, whereby there are no embryos left for transfer, and all of them are shown to be affected. Very often there are claims or cases whereby they show things like height or intelligence, or what you'd call extracurricular changes that you would make, and these are all polygenic and misunderstood entirely, so they would never really be done. It always frustrates me hugely that we do not consider that germline genome editing would not be treated in the exact same way as we treat pre-implantation genetic diagnosis and the introduction of every single new technology or new disorder that we treat for, all has to go through the human fertilization in the UK, at least through the Human Fertilization and Embryology Authority before it is approved. So every single condition is approved on a case-by-case basis. So the idea of this was an idea up until 2 years ago at the Second International Summit on Human Genome Editing, where the news was released that a researcher, He Jiankui, had in fact not just taken his experiments, or had in fact taken his experiments beyond experiments and into the clinic. Through a series of experiments, he had edited the embryos of a number of patients. He had recruited them to tell them that he was coming up with a vaccine for HIV. And what his plans were, which he did execute, were to edit a gene called CCR5, which when mutated, renders HIV incapable of entering into a cell. So his goal was to make these babies immune to HIV. So when this news broke at the eve of the international summit, there was a lot of backlash. First of all, they wondered whether the first claims were to say that actually he was saying that he did it and that he probably hadn't done it. I think for those of us who knew a little bit more, we thought it wasn't whether he had done it, but it was rather the extent to which he had done it. So what he announced was that 2 twin girls had been, or rather one pair of twin girls had been born with their genomes edited to mutate CCR5. There were all sorts of wrong things with this experiment. As much as I am talking very positively about the use of the technology at some stage, there were so many things wrong with this that it makes a lot of us angry for the backset that he's put in terms of other people's ability to use this technology just for research alone because of the major backlash internationally. First and foremost, he did not address an unmet medical need when he was editing a human embryo. There are plenty of antiretroviral therapies for HIV. The actual editing he did was very poorly executed. They weren't actually able to tell following the experiments whether or not they were heterozygous or whether or not they could actually contract HIV still. Nobody's going to expose them. It wasn't actually clear what the new mutations would do. And there were many, many problems with informed consent. So he was pretty much renounced by the scientific community and his peers. And yet there were documents to suggest that potentially his experiments had been funded by the Chinese government. This is him under house arrest. He has actually since been put in prison for these experiments. But it does make you wonder about some of the headlines that came about with the first IVF babies. So you can see from this very damning headline, "Do not work with this man," you would not equate this with the same person who has won a Nobel Prize. And this unfortunately was the premise of He Jiankui's, I guess, motivation, that he assumed that he may be pilloried at first, but that later on he would be rewarded for his work. And I am not in any way comparing the work of these two, but I'm just saying how different headlines can paint a very different picture in different times and scenarios, and they can change. So I think it's very important as a result to put this in context. We have, when we look at both gene therapy and pre-implantation genetic diagnosis, both of them have been attempting to do similar work to what germline genome editing would do. With gene therapy, we've spent over 30 years just trying to ameliorate symptoms or to try and compensate for a protein that is missing by the insertion of viral vectors that can be used as carrier vectors for a given protein. And equally with pre-implantation genetic diagnosis, where it is, as I mentioned, licensed on a case by case, it aims just to avoid. It involves the insertion of needles and the removal of genetic material. It's certainly not free from harm. But if we could, which we can, should we correct to cure? I think it really only takes a quick look at the number of even single gene disorders that affect how many people, 400 million people every year, for us to realize that this is something that actually we really need to address. And the timing is quite right at the moment with it being Rare Disease Day coming up. We are really fortunate tonight. Dr. Wametha did her PhD in the laboratory of Kathy Nyakin, who is the first person to have done genome editing on human embryos for research. And we are also joined later by a current member of her lab, who is also going to be talking about their work on editing human embryos. I will say that the fundamental thing that we need to know is how this works and how we can actually start to see whether and where it may go wrong. The initial headlines were to say that the genetic modifications could help researchers develop treatments for infertility, when really, actually, we just wanted to know fundamental information about how an embryo develops. So there have been plenty of safety concerns raised with germline genome editing. And I just want to very quickly say that when you think of a given experiment or outcome, you have to account for every different type of tool that has been used in the CRISPR toolbox. And as I mentioned, all of these tools are evolving by the week. So we have new methods of delivery, new modes of delivery, different types of endonucleases. Our guide designs are evolving quickly. So I think it's only ever giving you one time point and one given experimental outcome. So these are just some of the missing pieces of the CRISPR puzzle, all of which yield different outcomes in a given experiment, depending on what you are using. I call it the nucleotidal wave. It's very difficult to keep up with all of the new advances, but it is certainly exciting. It's really important to know that genome editing is not just being used for editing genomes. We are moving beyond that to the ability to edit the epigenome, to edit our methylation, to do base editing, which doesn't necessarily break the DNA, but actually fixes a single base with high resolution and targeting ability, and also to engineer cancer cells to enable them to overcome the immune system. So the risks of genome editing, I'm being really quickly, Sissy's turned on her camera, which makes me think I'm over time. The risks of genome editing are always that stated that you would have unwanted editing, that you may have off-target effects, that you may get what called CRISPR-derived mosaicism, that not every cell would have the intended correction, or that you may have a transgenerational change. And I just want to take a quick moment to take a comparable look at what the risks of assisted reproductive technologies are. Unwanted editing, I definitely don't think any embryo wants a single cell or a clump of cells to be removed. There have been plenty of papers showing the potential off-target effects of biopsy, of laser, of culture media in general. Mosaicism is the biggest topic and a never-ending drama in the IVF world, and is still consistently debated. And of course, the transgenerational change that we are witnessing one generation down the line after children who have been born from IVF and ICSI. So I think if we were to try and legislate for IVF right now, I think we'd actually really struggle. I just wanted to take a note to also have a look at the 1974 paper that James Watson said, where he foresaw all sorts of bad scenarios from IVF and its application. So the limitations are that it's a new technology relatively. Legislation does not move as quickly as technology does. I think there's a lack of human biological material that will enable us to actually do experiments. The only, human trial when it comes to embryos is human trial. And with that, and with an ever evolving technology like this, where it's getting incrementally, in fact, more than incrementally better by the week, it's hard to draw the line on saying when good is good enough, when we don't have enough research in this area. So if you're interested, please look at the Nuffield Council on Bioethics, and I will take your questions and move on. Thank you.